Hey guys, welcome back to biology. I am going to walk you through what we're going to do today. I have everything opened up that you'll need for today's class just so I can show you what that looks like on your end. But before we get into that, quick announcement and then the agenda, and then we'll get started. So we will have a quiz next class. This will be through Google Forms. You can use your notes on the quiz, but what you need to know is DNA structure, base pair rules, and replication. We talked about DNA structure before all of this happened, so if you don't remember any of that, feel free to pop into office hours and I can help you out. If you don't have your notes, I can send you a copy. Just shoot me an email. And then today we're going to talk about base pair rules and replication. So here's our agenda. You have your catalyst, which we didn't do, so let me just cut that out. So we're going to do some guided notes, and then you guys will have an exit ticket. So pretty simple day today. But I do want to walk you through what that will look like now that we're doing it in an electronic format. So these are the notes that you guys will open up. They will be linked for you in Google Classroom. You're going to hit File. You're going to go to Make a Copy. Title it whatever you want, and then you can type directly into that document. So I recommend split screening between our guided notes and then our slides. Once you've finished your slides and your notes, you're going to take a quick quiz through Google Forms. It's just got 5 questions for you. 2 of them are just identifiers. So this will be my way of making sure that you understood what we covered in today's lesson. So without further ado, let's go ahead and get started. All right. So for today, we are going to be talking about the steps in DNA replication. You guys will be able to model that process, and then you'll also be able to recognize the base pair rules and apply that as necessary. So we're starting off in the review part of your notes. That's the very beginning of the note sheet. When we're talking about base pairs, there are 4 bases that make up a DNA nucleotide. You have adenine, which we represent with an A, thymine, which we represent with a T, cytosine with a C, and guanine with a G. You won't be asked to remember how to spell these on a test, so don't stress about that, but make sure you recognize the different letters. The part that matters the most is the complementary base pair rule. What that tells us is that when DNA lines up, remember it has 2 separate strands, it's a double helix. If there's an A on strand 1, it's going to pair with a T on strand 2. If there's a C, it will pair with a G. So the way to remember that is that apples are in trees and cars go in garages, if that helps you with it. So to help get our brains flowing, you can actually do this in your notes. These are already typed in for you. So you have 5 different DNA base pairs or strands. You guys are going to figure out what the complementary strand would look like. So we've got number 1 here. I'm going to walk you through how to do this. If you already know how to do it, you can go ahead and start typing for 2, 3, 4, and 5 while I talk everyone else through. So if you look at this top strand, you've got all of these different bases that have paired together to create 1 strand of DNA. Our job is to figure out, what does the complementary strand look like? What is the one that's opposite of it going to look like? So if you remember the base pair rule, this process is pretty easy, right? We know that A pairs with T, so I'm going to write a T here. And the same is true for T and A, so it's just the opposite relationship. And we know that C pairs with G. So if we go down here to number 2, G is going to pair with C. So you put a C right here. C pairs with G, so you'd write a G right there. The process is pretty simple, and if you get confused by it, that's okay. That's what office hours are for. You can ask questions on this link. But I want you guys to get into the habit of doing this, because when we get into RNA replication, this will be important as well. So go ahead and do 2 through 5. You can pause the video. And then if you have questions on it, or if you want me to check it, I'll be looking at your notes on Wednesday. But you can also just shoot me a quick email and I'll check it out then. So that brings us to today's actual work. So this is our new content. It's labeled in your notes. You guys are going to be able to explain this process of DNA replication and organize the steps in order. This is a beautiful Microsoft Paint rendering of what that process looks like. I did not create it, but what I like about this is you can see We start with one strand, that's the blue one here, and it unzips or pulls apart. Then a new strand starts to form, that's the pink, and it forms the complementary pairs. So it creates, if this were A, then this would be T. If this were C, then this would be G. And it creates the opposite of that. So we're saving one half of the strand, that's called the semi-conservation model, and we're ending up with two identical copies, one with the half of the original strand and then half of the new on each. So DNA replication is the process by which we copy DNA during our cell cycle. Before we get into detail about what this looks like, I want you to go ahead and pause the video and think for a second about why this might happen. So DNA replication happens because we need the exact number and type of chromosomes in new cells. Remember, we talked about that when we talked about genetics. So if you change the number of chromosomes, you change what type of organism it is. We have to have the exact same type and number of chromosomes in order to get the same types of cells and organisms. If your skin cells didn't copy exactly, then you might end up with a liver cell where your skin should be. Right? Every new cell needs an exact copy of the DNA that it originated with. So this is your official definition. This is the one that you're going to type into your notes. And I did make this in Microsoft Paint. You're welcome. That was a lot of love of labor there. So when DNA replicates, it's going to make two DNA molecules that are identical. That means they're exactly the same to one another as the original that it started with. So we start off with one strand. Remember, it kind of pulls apart, and then a new strand is formed. That's what this purple represents here, to pair up with the original. Replication happens during the synthesis stage. Remember when we talked about the cell cycle, you've got G1, G2, and S. All of those are part of interphase. S happens between G1 and G2. That's our synthesis stage. And that's when the DNA actually gets copied. So the single strand acts as a template for the new one, and this happens through 4 different steps. When we're looking at these steps, I want you to pay attention to 2 things. First, what happens? Second, who does it? And when we say who, we're talking about which enzyme is responsible for facilitating the process. So our first one is unzipping. It's exactly what it sounds like. This is when the strand physically separates into 2 parts. So for the unzipping stage, our double helix is separated into 2 strands, and an enzyme called helicase is the one responsible for this. Remember, the ASE ending tells us that it's an enzyme. This helic part here tells us that it's separating the helix, right? It's separating the 2 strands. Now, the DNA gets unzipped in both directions. So helicase attaches to one spot, and then it runs in both directions to help speed up the process. Our second type is base pairing. So after the DNA strands have been separated, all of the different bases that match are going to start floating into the places that they need to be. Remember, whenever we're talking about chemical processes, you have molecules just bouncing around until they get to where they need to go. So an adenine base, might just pop around until it finds a T and then it attaches. The same thing happens with cytosine and guanine. So you have something called free nucleotides. These are bases, A, T, C, and G, that aren't attached to a DNA strand. They're just floating around in the nucleus. And once that strand separates, they're going to pair up with their complementary bases. Next up, we have our bonding stage. So it's exactly what it sounds like. This is when a new bond is formed using an enzyme called DNA polymerase. Remember, ASE tells us that it is an enzyme. The polymer part here, remember a polymer has many parts. So this is taking a bunch of different nucleotides and actually attaching them to the new strands. So our new base pairs, these guys floating around right here, are attached to the old strand to form a new double helix. So you can see we've got 2 new strands being formed. And then once all of that is done, polymerase has a second job to do. So this guy's pretty critical in this process. So once the DNA molecule has been created and bonding has occurred, Polymerase goes back over the strand and proofreads it, kind of like you guys would proofread an essay before you submit it to Mr. Hansen, hopefully. I don't know if you actually do that, but you should, because he can be critical, right? So when we create new DNA molecules, you have to check over those to correct any errors. If this correction process doesn't happen, that's when we end up with mutations. Some mutations are beneficial, but those are only usually ones that we're born with. The ones that we get over time tend to cause things like cancer. All right, so polymerase is critical in making sure that those have bonded properly. What we end up with as a result, we've already talked about before, are two exactly identical DNA molecules. So each one has one of the original strand, that's the blue, and one of the new strands, that's the orange. Both of them have those complements that we talked about with the base pair rule. This is an image of what the whole process looks like for you. So we start off with our parent DNA. Step one, remember we unzip. Helicase runs up this guy in both directions and starts to separate those pairs. The bases are held together by hydrogen bonds, so it's a pretty weak electrostatic bond, and that's why helicase can so easily separate them out. After that, our complementary base pairs, our free nucleotides, start to attach, and then the bonding occurs. After they're bonded together, remember polymerase does its final step, which is proofreading, and we end up with two identical molecules. This is just a quick animation, to show you what this process looks like.
To make perfect copies of our DNA, an enzyme first unwinds the two strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine.
All right, so that's our second and third step. The free nucleotides come along, and then DNA polymerase starts to attach them.
In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
Oh no, don't do that. All right, so that just gives you an idea of the basics of this process. We're going to get into more detail as we go through this unit. But for now, your next step is going to be to complete your exit ticket. It says silent independent. I don't care if it's silent, but please do it independently, right? So make sure that you're not using your notes. I want you guys to see what you actually know so you can figure out what you need to review before any quizzes or tests. Remember your steps for today. You're going to go here. After you've filled in your guided notes, you're going to submit those on Classroom in this post. So on the Google Classroom post, the notes will be attached. You make a copy, you type it in, you upload your notes. The other thing that you need to do is on your exit ticket, you're going to fill in that information and submit it to me. And then finally, we're going to pop over to Classroom just so you guys can see the final step. This is more for attendance purposes than anything else. So if you forget, I understand, but please make sure that you're doing everything you can to click this turn it in button every time you do your work. So when you go to a post, let's go to my classwork for prepping for PD. Right, teachers had to make an office hour and advisory link. When you click view assignment, it has a space for you to put your work over here. You're going to hit that add or create. You're going to put your Google Drive file that you just created on there. And then the most important step for me is after you finish it that you click mark as done. Make sure you're both uploading it and you're clicking mark as done or turn in work so that I know that you finished those assignments for me. Again, feel free to email, text, comment on Classroom. Between 9 and 11 is when I'll be available to answer questions directly on Classroom. But I'll also be free from 2:45 to 3:45 p.m. for you to come to office hours. That will be linked, and I'll pull it up to the top of our feed on Classroom. around 2 p.m. just so you have that available to you. Thanks, guys, and it's good to see you again. Let's get back to it.